In our next Genetics and Biotechnology, we're going to be looking at Lesson 6, How does DNA code for traits? So as we know, we have been following along with the Miller family storyline, and we know that Mary did genetic testing to see if she was at risk for having hemochromatosis like her brother David. And so we took DNA samples from Mary, but also from Jack and Jill, her parents, Jack and Jill Miller. And the testing for hemochromatosis, remember, involved looking at the sequence of a very specific gene called HFE. So we have already explored the DNA strands of Jack and Jill. We have already now explored the RNA strands, and so we're going to take it to the next step where we see which of the amino acids are produced from the RNA of Jack and Jill Miller. Okay, so how does DNA control our traits? How does the information carried in a DNA sequence impact the outward appearance of an organism? Well, if we think back to the central dogma from the DNA recipe to the RNA copy to the protein product, we know that DNA is in the nucleus of the cell and that the DNA gets copied onto a piece of messenger RNA, and the messenger RNA leaves the nucleus of the cell and goes out into the cytoplasm where it finds the ribosomes, and the ribosomes are where the proteins are made. So DNA carries the instructions for building proteins. The proteins are made of long chains of amino acids, and these proteins are what control how an organism looks and functions. So what are amino acids? Well, there are 20 amino acids found in living things, and the order of amino acids plays a role in the three-dimensional shape of the protein. So we have, based on our DNA and RNA, we have what's called a protein polypeptide chain. And all those little amino acids form like a code. And those amino acids are what's going to cause our cells of our body to act certain ways or to display certain traits. Now, in this lesson, we're going to use these little children's pop beads. You may have played with some beads like this when you were a child, but we're going to use these big pop beads as our amino acids. Okay? Now, what is a codon? Now, we know we're going to be looking at these amino acid polypeptide codes, but what is a codon? Well, a codon is a 3-nucleotide sequence. So that's where we take the sequence and we divide it up into groups of 3. So the first 3, like AUG for example, would be codon 1, ACG, codon 2, GAG, codon 3, and so forth. This is just an example. Your DNA could be in a different order here, but this is just an example. A codon, though, is a 3-nucleotide sequence. So every 3 of those nucleotides or those nitrogen bases form what's called a codon. So AUG might code for some amino acid that's different than ACG, or GAG may be different than CUU, and so forth. So all of these little amino acids are going to be slightly different. Okay? So on paper, in Lesson 4, we created our complementary sequence to DNA. Then in Lesson 5, we took that complementary DNA sequence and made the RNA sequence. And now we're going to use those codons of the RNA sequence to find out what our amino acid sequence is going to be. Okay, so on our RNA, if we had the order of codons AGA, then that's going to cause the ribosomes to make an amino acid called arginine. And arginine's symbol is ARG. If the RNA has the code UGC as one of the codons, then that would cause the ribosomes to make an amino acid called cysteine, or CYS. If the RNA sequence is GAG, then that would cause the ribosomes to create an amino acid called glutamic acid, or GLU. If it has CAG, then the ribosomes are going to make glutamine, or GLN. If the ribosomes get the code CAC, then they're going to make the amino acid histidine. If they get CUG, they're going to make leucine. If they get ACG, they're going to make threonine. If they get UAU or UAC, they're going to make tyrosine. And if they get GUG, they're going to make valine. So as you can see, each one of these amino acids is going to be represented by a different pop bead. We have the green smooth barrel beads. We have the blue wavy bead. We have the blue diamond-shaped bead. We have the green wavy bead. We have the green diamond bead. We have the yellow smooth barrel. We have the red barrel that has the indented ends. We have the yellow wavy bead, and we have the red wavy bead. So those are going to represent the amino acids from the codons. So to assemble our codons in a proper order, right here we have GILs A1 and A2, an example of them here. Here's what the RNA for GILs A1 and A2 looked like. And here's what the amino acid sequence with the pop beads looks like. So we're going to make that for Jack and for Jill, and then we're going to compare Jack and Jill's bead models. Okay, so here's going to be the results. So spoiler alert, here's what our results are going to be. If Jack or Jill has this particular set of beads, exactly like this right here, then they have the typical HFE gene, which is normal and would not carry hemochromatosis. But if they happen to have the HFE gene that looks like this, then they would carry the trait for hemochromatosis. So as you can see, there's just a slight difference. The first one is the green smooth bead. The second one is the yellow wavy bead. Third, we have the red double barrel bead. Then we have the difference right here. In the normal HFE gene, you would have cysteine. But in the HFE gene that has the mutation for hemochromatosis, we would see the tyrosine. Then we continue on. The green wavy bead's the same. The red wavy bead's the same. The blue diamond is the same. And the green diamond is the same. So as you can see, it's just this one little difference right here between cysteine and tyrosine that causes a world of difference in the DNA of this family. So if your Jack or Jill have the typical HFE gene, then they would not have passed on hemochromatosis. But if they even have one copy of the HFE gene with the mutation, then they could have passed on hemochromatosis to their offspring. So in order for their son that has hemochromatosis, David, in order for him to have hemochromatosis, then he had to have inherited the HFE gene from both parents because it is recessive. So it's not going to show up very often. And in order to get the recessive trait to appear in a person, then they would have had to have received that recessive trait from both parents. So we know that both parents are carriers of the HFE gene with the mutation. So we will be filling out our genetic testing result for Jack and for Jill Miller. And we see that both of them have the mutation. So Jack Miller has one mutation. So he is a carrier for hemochromatosis, which means he does not have the disease himself, but he passed it on to his offspring, or could have passed it on to his offspring. Jill Miller also has one mutation, so that means that Jill Miller is also a carrier for hemochromatosis. And so we use big H, little h to represent the hemochromatosis. The big H is the typical gene, and the little h represents the mutated gene that causes the sickness. So what are the chances that Mary inherited the hemochromatosis gene from her parents? We know that Jack is big H, little h, and Jill is also big H, little h. So when we do the Punnett square for them, we see that there is a 25% chance that their children would be unaffected. There is a 50% chance that their child would be a carrier. And there is a 25% chance that their child would be affected with the hemochromatosis gene. So Mary has a 25% chance of having hemochromatosis, which sounds kind of small, but in this family, we already know that David has hemochromatosis, so we know it is possible. So based on what you know about hemochromatosis and David's health, what would you expect David's bead models to look like? His would be the mutated genes from both parents. As you can see right here, he has the yellow bead for tyrosine instead of the blue wavy bead. So here's the rest of the story. 1 in 10 Caucasians is a carrier for hemochromatosis. So how did this disease become so common? If you would like to find out more about hemochromatosis and how this disease became more common, you can go to the Hudson Alpha website, or you could also do a Google search for hemochromatosis. Now, let's take a look at our textbook or manual for the genetics and biotechnology. You need to make sure that you read lesson 6. As we scan through lesson 6 here, you will read about translation, about the codons and amino acids and the proteins. You also read about inheritance of hemochromatosis. It gives a picture of the codon chart. Then the protocol that the students will follow in class in order to build the model, the bead model. And then we come to the evaluate your learning, which is what you will be doing on your CAMI document. So if you open your CAMI document, you have a few questions to answer from the reading passage. You do also have a career connection where you can explore the career of being a genetic counselor. Then we have our results pages down here, where you can use the results in the video to fill out the testing result for Jack and Jill Miller. Okay, to recap our lesson, you should watch this video. You should open the Genetics and Biotechnology manual right here on this link, read lesson 6, and then open and complete the assignment sheet with CAMI.